Sir, it's
Hello.
sir. Now you're live. First, I introduce you, then you can start.
Okay. Okay.
Good afternoon to all. I, on behalf of Institution Innovation Council, Tripura University, welcome everyone in this workshop on screening natural products with antimicrobial potential and cytotoxicity screening. We are privileged to have with us Dr. Shrujit Bhattacharjee, Assistant Professor, Department of Molecular Biology and Bioinformatics, Tripura University, to enlighten in this topic. I welcome you, sir. Now over to you.
Okay. Thank you, Ajay. And today we are organizing an workshop on screening of natural products for their antimicrobial potential and their cytotoxicity activity or their cytotoxic potential. And this is the part of Institutional Innovation Council activity of the Pune University. Today, we are arranging this workshop to give a brief idea how we can screen the natural products for their antimicrobial potential, as well as once we have screened out a potential antimicrobial agent, before further proceeding, we need to check their cytotoxic activity so that we can use them safely. So why we need antimicrobial agents? Starting from the development of the first antibiotic, penicillin, which is developed by Alexander Fleming in 1928, there are hundreds of new antibiotics has been evolved, but due to inappropriate use, coupled with the shortage of the newer types of antibiotics, there are large numbers of antibiotic resistance phenomena has been developed, and to overcome these kind of antibiotic resistance, we need to identify a new compounds or new agents which can be resourced or which can be originated from the natural resources. And in this regard, herbal medicine which we obtain or the natural products which we obtain from the natural resources like the plants and the other natural resources, we can use them or we can screen them for their potential antimicrobial potential, for their potential antimicrobial activity. And contrary to the synthetic drugs, these antimicrobial substances which we can obtain from the natural resources, mainly from the plant sources, mainly from the which are mainly from the plant origin, cannot are not generally found to be not associated with the side effects, and they have an enormous therapeutic potential to heal many different infectious diseases. And there are plenty of reports where it has been shown that plant extracts from higher plants have or possess antibacterial, antifungal, and insecticidal properties under the laboratory setup. And here we can use different methods to screen the antimicrobial activity of an new agents or of a natural products which we can extract from the natural resources. And one of the simplest method is to characterize the minimum inhibitory concentration, which is the smallest concentration of the compound agents or antibiotic which can inhibit the growth of organism. And the MIC can be determined by using the liquid or called broth dilution method, or we can use the solid media which is called the diffusion method, or we can use the e-test. We can use both of these tests allow us to determine the MIC. In addition, for the quick screening, we can use the beta lactamase production for the screening of antimicrobial potential of natural products. Now, uh, the here we are. I am going to describe a brief about the broth dilution method. The main aim of the broth dilution method is to determine the concentration of the antimicrobial agents which we want to screen for their activity under the defined test condition. And here the defined test condition means we can define the time frame, we can define the concentration, and we can also define the other parameters of the medium. And by using the broth dilution method, it helps us to visualize the growth of bacteria to drugs and allow us to evaluate the activity of new antimicrobial agents, which I have already described. The Whereas we can, instead of broth dilutions, we can also use the agar dilution, where we can incorporate different concentration of the antimicrobial substances into the nutrient agar, followed by the application of the standard number of the bacterial cell on the surface of the agar plate. And for broth dilution, which we generally carried out in 96 cell microtiter plate, where the bacteria are inoculated into the liquid growth media in the presence of different concentration of antimicrobial agent. And then growth is assessed after the incubation for a defined time frame, that is generally 16 to 20 hours. It can be extended up to 24 48 hours. And then MIC value can be determined by the spectrophotometric reading. And the these protocol are generally applied only for the aerobic bacteria, and it can be completed within 3 years. And it is the basic setup for starting the broth dilution method. Here, the it is the first one I have shown here is the tube method, where the tube containing increased antibiotic concentration, and then the bacteria has been inoculated. And after incubation for specific time frame, it may be 24 hours or 48 hours or 20 hour 18 hours. The rec- After that, the broth dilution okay, after the specific time frame of incubation, we can take the OD under using the spectrophotometer. And here, the dilution method we have used is a twofold serial dilution, that is starting from the initial stock concentration, we are diluting every time two folds. And generally, during the broth dilution method, we use 5 into 10 to the power 5 colony forming unit. It is the standard number of colony forming unit which we generally inoculate at the initial incubation frame. That is, when we incubate the bacteria with the compounds, we generally starts with 5 into 10 to the power 5 colony forming units per ml. But this method is little bit tedious and it takes longer time frame. And uh, the here it is a 96 cell microtiter plate. And here I'm giving you an example where we have used a 96 cell microtiter plate. And here, it is clearly visible that the depending on the concentration of the antimicrobial agents, the growth of the bacteria or the turbidity of the medium was changes with the different concentration. And here we have added, first we have added the medium, and then we have added the drug or the compound at different concentration by using the twofold serial dilution method. And then we have inoculated the bacteria at a concentration of 5 into 10 to the power 5 colony forming unit. And then after 48 hours we have taken the OD by using the microplate reader. And then we have in uh, collected the data, and based on the data we can determine the MIC value, which we have already discussed, that is the minimum concentration at which the growth of the bacteria was completely nil. Now, uh, in addition to the broth dilution method, we can also use the disc diffusion method, where the paper disc containing a defined quantity of antimicrobial agents can be placed on the agar medium, where the bacteria are being uniformly seeded. And depending on the concentration gradient of the antibiotic or the tested agents, these agents start to diffuse from the disk to throughout their periphery. And if the compound is inhibitory in nature, that is, if they are antimicrobial in nature, then until along with their concentration gradient, the growth of the bacteria was also found to be inhibited. And based on the distance from the disk to the clear zone in the medium, we can determine the susceptibility, or we can determine the inhibitory activity of the compounds on the tested organism. Here is the basic methodology which we generally follow. First, we inoculate the plate with the liquid culture media, and then we seed it uniformly throughout the medium, and then we place the disc containing the compounds at a defined concentration, and then we incubate it for 24 to 48 hours. And then the if the compounds has antimicrobial potential, then they generate a clear zone surrounding the disk, and based on the diameter of the clear zone we can determine the antimicrobial potential of the compound. Here we have carried out some of some of experiments. Here uh, we can clearly see that here we have used 4 discs where we have added the concentration, we have added the particular specific compound like called plumbagene at a defined different concentration, and then 24 hours of incubation we observe that plumbagene generates a clear zone surrounding the disk where it clearly inhibits the growth of the bacteria, and from that we can clearly define that plumbagene has a antimicrobial potential. And then uh, once we screen the antimicrobial potential of a compound, we can check the cytotoxicity potential or the cytotoxic activity of those compounds, because once a compound has been identified that it possesses antimicrobial activity, before further screening for their antimicrobial or anti-infective potential in a living organism, we need to check their cytotoxic profile, because if it is cytotoxic then it cannot be used in a living organism, or else it also creates a detrimental effect as it is toxic. So before going into further testing, we are generally test the cell viability or the cytotoxic activity of the agents which we generally test for their antimicrobial potential. There are different tests which we can use for checking the cytotoxic potential of a compound. The Here in the first panel we have shown the different principle based on which we can develop the cytotoxic assay. The one is based on the membrane integrity, which is called the Trapan blue dye exclusion method. The another one is called dye uptake assay, which is also based on the membrane integrity, which is known as diacetyl fluorescent uptake activity. And then we can use the radioactive method where the label chromium uptake assay can be used. We can use the enzyme released assay where the LDH, based on the release of LDH from the cell, we can determine the cytotoxic effect of a compound. Based on the apoptosis, we can also use the annexin V FITC for checking the apoptotic potential of a compound. And the one of the simplest method, which is colorimetric based method, where we can use MTT assay to check the cytotoxic effect of a compound. And here the basic principles has been given, and in most of the cases the all of them help us to measure the cell viability and cytotoxicity at a very short time, and it helps us to identify the dead versus live cell with a specific time frame. At many times when the cells are subjected to drugs or the other compounds, the effects are not immediate, but it requires several hours or more than days to observe the effect. Now, during those cases, we we cannot use the assay which requires which are immediate type, like the trap and blue dye exclusion method. And here in our laboratory, we generally use the MTT assay to check the cytotoxic effect of a compound. By using MTT assay, we can determine the number of viable cell in a medium or number of viable cell in a culture condition. And MTT it is a colorimetric assay for assessing the cell metabolic activity. For determining the mitochondrial dehydrogenase activity in the living cell, NADH reduces the tetrazolium salt of the MTT to an insoluble formagen, which gives a purple color. At a normal condition, if a cell is in living state, then only it has the ability to reduce the MTT to convert it into the insoluble formagen. Whereas if a cell dies, then it loses its ability to reduce the tetrazolium salt. So that's why MTT can be used to determine the survivability of the cell, or in the other way, the less the survivability, more is the toxicity. So in this way, by using MTT assay, we can determine the cytotoxic potential of a organism or cytotoxic potential of a compound. Here we have carried out MTT assay using the plumbage in a different concentration. And here we can clearly see that when we have used the high concentration of plumbagine at the in the middle column or in the middle row, it is clearly seen that it more formagen has been produced, and where the cytotoxicity is less. Whereas at a lower concentration, it is found that the penetrability of plumbagine increases, and which increases the cytotoxicity of that compound. So it help us to determine that plumbagene cannot be used as a single or compound to be for the antimicrobial activity, or we cannot use plumbagene directly into the animal system. Instead of that, what we have done, we combine plumbagene with the other compounds to minimize its cytotoxic effect. In addition, we have also prepared encapsulated plumbagene for their targeted delivery. Now, in addition to the MTT, we can use the related tetrazolium salt like XTT and MTS. The XTT, the main difference between these three compounds, the MTT is insoluble purple formagen, whereas the XTT is a water-soluble product, and its sensitivity is higher. Whereas the MTS, it is also a water-soluble products, but is absorbance maximum at 490 degrees, 490 nanometer. But the MTT, the main problem with the MTT, it is insoluble formogen, and for solubilization, we need to add either acetic acid or the other or DMSO to solubilize the MTT. In addition, it was also observed that MTT itself is cytotoxic. Therefore, uh, in in our laboratory, we have started using the XTT for as an alternative to MTT. And the main application of MTT, we can use it for to check the cell viability. We can also measure the cytotoxicity. That means when the viable cell is low, that means it is more cytotoxic. We can use for the drug screening on the cell lines and the patient samples, and we can determine the IC50 value, that is inhibitory concentration 50, that is at a concentration at which the cell viability become only 50%, or cell the loss of cell viability becomes 50%. And these are the basic equipments or the materials which you require for to carry out the MTT assay or the uh, other XTT or MTT or XTT assay. Like we need 96 microplate, we need 37-degree carbon dioxide incubator, we need the samples, that is the drugs or the tested compound, we need ELISA reader, we also need cell culture medium where we can culture the cells, and we need a multi-channel pipette because here need to uh, uh, we need to add the compounds at the same time in the different wells. And so by using multi-channel, we can increase the efficiency of of carrying out the serial dilution. And in addition, we also need the MTT powder, and we need DMSO as a solubilizing agent. And for checking the cellular viability under the microscope, we need an inverted microscope. And we have already discussed that MTT is cytotoxic and harmful, and MTT is light sensitive. Hence, during the time of experiment, we need to keep the plate wrapped up with the aluminum foil or other, or we need to keep the plate in the dark room. And to optimize the assay condition, first, we need to optimize the cell count, and also we need to optimize the incubation period for the specific cell line with the tested compound. And then we harvest suspension cell by centrifugation, and adherent cell, in case of the adherent cell, they can be released from the substrate by trypsinization or scrapping. Whereas for serial dilution, we have already discussed in the culture medium, we can use the multi-channel pipette, and for incubate the cell under appropriate condition, we can incubate them from 6 to 48 hours. And then we can add 10 microliter of the MTT reagent to each well once the incubation with the tested compound has been done. And after that, we can return plate to the cell culture incubator for 2 to 4 hours. That is, after adding MTT, we need to keep the plate at least 2 to 4 hours in incubator by wrapping up with the aluminum foil, or we need to keep them in the incubator in a at the dark place. That is, it should be kept in dark place because we have discussed that MTT is light sensitive. When the purple precipitate is clearly visible under the microscope, we need to take out the plate and we need to observe it under the microscope. If we observe that there are purple precipitate, then we need to add the 100 microliter of detergent reagent, which can solubilize the MTT. And then we need to leave the plate with the cover at the dark again for 2 to 4 hours or overnight at room temperature. And then we can absorb the plate under the microplate reader at 550 to 720 nanometer wavelength, and then we can prepare a standard curve by plotting the number of cells on the x-axis and the absorbance on the y-axis. And the number of cells with the linear portion of the plot yield a absorbance of 0.75 to 1.25 is suitable. That is, the absorbance rate should be 0.75 to 1.25. And then these are the precautions we need to take to when we are carrying out the experiment. That if the cells are in suspended condition, then we generally prefer to use the V bottom plate. Whereas if the time from starting incubation to taking measurement is over the 48 hours, it is necessary to exchange the media, or rather to remove some part of the media and add fresh media. And then the during the removal of the media, we need to tilt the plate so that the tip cannot touch the cell at the bottom of the plate, or at the but that means we need to minimize the contact between the tip and the cells. And then for the floating type of cell, we need to centrifuge before removal of the media, so that the suspended cell can be pelleted down at the bottom and we can remove the media from the top. And the as we have discussed, that exposure time depends on the test substance, substances or the drugs which we have tested, and the purpose of the experiment. If the substance is highly toxic to the cell, then we need to expose them for a short period of time. Whereas if the substance are found to be slowly affect the cell function, then we need a longer exposure time. So that's why we need to carry out a standardization experiment. And this is the basic experimental steps which we generally carry out when we are doing the MTT assay. That is, first cell culture in 96 cell plate, and the preferable number of cells is 1.1 into 10 to the power 4 cells per well. And then we incubate it 24 to 48 hours at the CO2 incubator, and then we treat the drug or the compound at a variable concentration, and then we incubate it for 6, 12, or 24 or 48 hours as per the requirement or as per the specificity of the compound tested. And then we add 10 microgram of the MTT at a concentration 5 mg per ml reagent, and then we are going to mix the media with the MTT properly and in a homogeneous manner, and then we incubate it for another 3 to 4 hours in at CO2 incubator at 37 degree. And then we dissolve the formagen, that is the formagen which is formed due to the reduction of the MTT, and then we kept it for 30 minutes in the dark room, and then we measured the absorbance at 550 to 720 nanometer wavelength. And it is the basic protocol we have discussed, that if when MTT has been added, the cells which are viable, they convert the MTT to a purple formagen, and these purple can be visualized by dissolving the MTT or the converted formagen into the DMSO, and then we can take the OD in a specific wavelength. And for calculating the cell survival by using MTT, we generally use this formula, that is, survival rate is a sample where the drug has been used and the control where we did not add any drug. And then by using this formula, we can calculate the survival rate. And here, if we have a background absorbance, that is when we generally add sometimes MTT in a blank medium, depending on the blank absorbance to remove the background effect, we can use this formula, that is the OD from the sample minus absorbance, and from the OD in the control set minus absorbance, and into 100. So this is the formula which we generally use when we have a background absorbance. And if we don't have any background absorbance, or rather if we negate the background absorbance, then we can use the above number one formula. And the relative ratio, that is the ratio between the sample and control, we can determine by using this formula, that is sample by control into 100. And the true relative ratio gives us from the by removing or by negating the background observance. The absorbance is a background where we have discussed that where we did not add any cell, and we are taking OD from that only media. And it is the concentration here. A typical graph has been plotted where with the increasing concentration the cell survivability has been decreases. So based on this we can determine the effect of the compound on the cell survivability. And by using the MTTS, we can also determine the IC50 value. And these IC50 is the, we have already discussed, it inhibits the 50% growth of the cultured cell or the tested cell. And MTT help us to determine the IC50 value of the compound. And by using the above method, that is by serial dilution or the disk diffusion, and along with the cytotoxicity, so far in our laboratory we have tested a large number of compounds, and among them these are the compounds which we found to be suitable to carry out the further uh, anti-infectivity assay, where we have designed the experimental strategies by which we can carry out the experiment to test their uh, efficacy against different types of bacterial related infection. And we have already tested the some of these compounds like Vitaxin. It shows a potential antibiofilm activity, and it gives us a very good result where we have found that it prevents the biofilm associated infection, and it also helps the to prevent the growth of the microorganism at the site of wound. And we are carrying out the similar experiment with the other compounds. And uh, these are the, and one interesting compound is the plumbagin, which we found is, although it is toxic, but we are trying to reduce its toxicity by keeping its efficacy as an antimicrobial agent intact. Thank you. And if you have any question, please, the presentation is open for discussion.
Thank you, sir. Thank you so much for your words. I did not see any questions in the chat box, so we are conclude here the workshop. So once again, thanks to you.